My topic is comparative in-vitro therapeutical and nano-biological study of textile and endophytic fungus called Lantana camara, collected from the tribal region of Odisha state, India. So this Lantana camara is a white plant that grows in the wild, and it has a high
Sorry to
potential of
interrupt you,
pharmaceutical
Dr.
value.
Sahoo. Can
Sorry.
you, sorry to interrupt you. We cannot hear you clearly. Maybe if you can be closer to the microphone.
Am I audible right now?
Perfect. Thank you so much.
Okay, so my topic is comparative in-vitro therapeutical and non-biological study of leaf extract and endophytic fungus from Lantana camara collected in tribal region of Odisha state, India. So basically this Lantana camara, as you can see in the picture, is grown in the wild and has been the one of the least explored plant for the pharmaceutical and therapeutical use. So in my subject, I have gone for the therapeutical biotherapeutical study along with the potential for synthesis of silver nanoparticle and their application in biotherapeutical prospect. So the background study is based upon provide some insight to new age herbal medicament with importance of microbiological synthesis and modification of plant extract and green synthesis of silver nanoparticle where they can be used in relation to development of new pharmaceutical drug component that are more effective, safe, reliable, less or no toxicological effect. So my research design is based upon first collection of the plant sample along with the processing of the plant sample with respect to different studies such as qualitative estimation of different phytochemicals, quantitative estimation of flavonoid, alkaloid, phenolic and tannin, in vitro analysis, FDR and expense analysis, morphological study of SCM and PM, and the subsequent in vitro analysis of aqueous extract and silver nanoparticles that have been produced by the extracts. So, my materials and methodology, the plants that was collected was Lantana camara that was collected from the tribal region of Koraput state in India. And the plant material was set dry, especially the leaf sample and the root sample that I have collected for 8 to 10 days to reduce the moisture content. The dried plant was made into powder by mortar and pestle, and the powder was then sieved to powder form and kept in the zipper pouch for further analysis. Extract preparation, the plant material powder of Lantana camara was placed in amber colored bottle and some solvent was used that was water and ethyl alcohol in the ratio of 55:45. So the type of extract that we have got is hydroalcoholic type. The sample and solvent were taken in 1:20, means 1 gram of sample to 20 ml of solvent. The sample solvent was kept for 48 hours so that the solvent can extract all the phytochemicals, phytochemical risk from the leaf sample. After 48 hours, the mixture was filtered and the filter was collected. So the filter was desiccated by rotatory evaporator to get the condensed extract. So ultimately, that condensed extract was then dissolved in DMSO at a final concentration of 0.1 mg per ml. So in the right side in the picture, you can see the solvent extract, that is the hydroalcoholic extract that has been accepted from the leaf sample of Lantana camara. So for preparation of some types of extract from endophytic fungus that is associated with root of Lantana camara. So first the metabolites were isolated by culturing the endophytic fungus in PDA. Actually the fungus was isolated from the root. So I used PDA as the media for supplement that is supplemented with 0.1 chlorochemical antibiotics. So chlorochemical antibiotics will reduce the growth of the bacteria. So some of the parameters for fermentation or production of metabolite were pH equal to 5.9, temperature equal to 30 degree, and the speed of agitation was 70 rpm. After incubation for 10 days, the broth was filtered by Wattman number 1 filter paper, and the filtrate was collected for the further processing. The filtrate that was collected was put in the separating funnel along with ethyl acetate. So there will be two phases of formation in between the aqueous and the ethyl acetate. So we have to take the extract, that is the aqueous of organic top layer, that is the ethyl acetate part. So for resultant identification of the endophytic fungus that we have secured from the root sample. So we have got cladosporium species. We have gone for the morphology on the PDA plate along with the LCBC or the lactophenol cotton blue staining. So qualitatively analysis of secondary metabolite was the first step, where the hydroalcoholic extract of leaf contains alkaloid, flavonoid, phenolic, carbohydrate, protein, glycoside, saponin, steroid, quinone, tryptophan, cyproterpene, and tannins. The esterified extract of endophytic fungus isolated from the root of the plant contain alkaloid, flavonoid, phenolic, tannin, coumarin, resin, protein, and glycolides. So coming to the quantitative estimation of four different types of phytochemicals, that are alkaloid, flavonoid, phenolic, and tannin. So these are of high pharmaceutical or pharmacological prospect. So as we can see, the alkaloid was highest in case of hello. The alkaloid was alkaloid, flavonoid, phenolic, and tannin were being estimated by using certain standards such as atropine, quercetin, gallic acid, and tannic acid respectively. And you can see the quantity is estimated by different methodologies that has been described in the table. So in regards to the result, so my first prospect was that with the green synthesis of silver nanoparticles by using the extracts. So 1 millimolar of silver nanoparticles, silver nitrate solution 4.5 ml, and leaf extract 0.5 ml was taken, and it was kept at room temperature with constant shaking. And after 36 hours of incubation, so the color of the solution turned to reddish brown, indicating the formation of silver nanoparticles. So here you can see both the hydroalcoholic extract and esterified extract that was extracted from the endophytic fungus have the potential for green synthesis of silver nanoparticle from silver nitrate solution. So here purification was carried out by a continuous centrifugation process, and characterization of silver nanoparticle was done by using spectral analysis, where we got the peak in between 420 to 450 nanometer that correspond to the formation of the silver nanoparticle. So in vitro analysis of antimicrobial assay was also done for the extracts along with the purified silver nanoparticle and extract plus silver nanoparticle, where from the tabulation you can clearly see that there is an increase for the increase in the activity of the silver nanoparticle and extract as compared to the purified silver nanoparticles or the extract alone. So in vitro analysis for antioxidant activity was also found out that the highest activity was in the hydroalcoholic extract, but whenever it came to hydroalcoholic extract plus silver nanoparticle, there was increase in the perspective of antioxidant property. So based upon the above result, so we have gone for the detection of IC50 value, where the percentage was 72.5% with RNA. So for the result of anti-inflammatory activity, here also the hydroalcoholic extract gave the highest activity amongst the esterified extract and AGNP, whereas in case of hydroalcoholic extract plus AGNP gave a better result as compared to the extract or purified AGNP alone. So this is the FDR spectroscopy of crude hydroalkalic extract, because crude hydroalkalic extract has given better result for the antioxidant and anti-inflammatory and the antimicrobial activity. So I've gone for the FDR spectroscopy, where there is presence of some aromatic and some aliphatic rings, along with some NH bond, CO double bond, C double bond, C, NH, etc. So this is the Hplc analysis, where the mobile phase was isocratic type, and the mobile phase that was taken was methanol, water, and formic acid, and the column type was C18 with a flow rate of 1.5%, 1 ml, and column type is 25 degree. Detector is 230 nanometer, and as you can see, there are 24 different types of peak that are present in the hydroalcoholic extract from the HPLC. So from the above result, for the purified silver nanoparticles was gone for the TM analysis, where the morphology of the silver nanoparticle was highly variable, and the assemblies were found to be aggregate for silver in the range of 15 to 20 for hydroalcoholic extract and 20 to 70 nanometer for esterified extract. So the conclusion is that natural derived active compounds have evolved more than a year and have unique chemical diversity that result in varied biological and pharmacological effect. This resource has become an integral part for development of new drug lead compound and scaffolds. Natural products play a pivotal role in drug discovery for treatment of human diseases, especially the critical diseases. Herbal-based drug system has richer source of data from clinical observation, clinical experiment, dose and doses that can give insight to safety and efficacy of drug system. The herbal drug system has better safety efficacy, less known side effects, biocompatibility, bioavailability, better cultural acceptability, easy metal availability that made the drug system a new dimension in modern world of therapy. Herbal extract and plant also have the potential for synthesis of silver nanoparticles. They can be more eco-friendly, greener technology, and efficient as compared to other physical and chemical method of synthesis of nanoparticles. Integration of nanotechnology to the therapeutical prospect. So increase in potential thereby concluding that this technology can be better explored for more efficiency, safety, and potential. Thank you.